The phosphoinositide 3 kinase protein kinase B mammalian target of rapamycin or PI3K-AKT-mTOR signaling pathway is crucial for the control of many cellular functions such as growth, proliferation, and survival. Its components usually present mutations that are associated with cancer, being particularly common in breast cancer. Around 60% of tumors harbor genetic alterations that hyperactivate this pathway. Currently, studies are limited to experimental analysis of inhibitors of the pathway on cancer cells. For this reason, in this study, our objective was to perform mathematical models of the PI3K-AKT-mTOR signaling pathway, including the drugs everolimus and buparlisib. Both drugs are beginning to be used for the treatment of breast cancer. Everolimus is a mammalian target of rapamycin inhibitor, while buparlisib is a phosphoinositide 3 kinase inhibitor. In this way, we were able to observe how these drugs inhibit the dynamics of the pathway by decreasing the activity of its components. For the dynamic modeling of the signaling pathway, it was required to obtain the experimental values of the kinetic constant of each pathway reaction, which describe the conversion rate of one component into another in time. The values of the inhibition constants of the drugs were obtained by bibliographic review. With that information, we were able to perform the mathematical models by using ordinary differential equations. They were written using Python programming language syntax. We analyzed the variation of the pathway components through 60 seconds using the range-Coda numerical integration algorithms of Tellurium program. Our results showed that everolimus has a stronger perturbative effect on the dynamics of the phosphoinositide 3 kinase protein kinase B mammalian target of rapamycin signaling pathway than buparlisib. With the inclusion of everolimus, the activity of the mammalian target of rapamycin complex 1 decreased by 33% and the activity of ribosomal S6 kinase by 2%. Both components played an important role in cancer because they regulate protein synthesis and growth of cancer cells. Buparlisib was shown to be effective in decreasing protein kinase B and phosphoinositide 3 kinase activity, but an increase in protein FOXO activity was observed, which could cause the activation of the mitogen-activated protein kinase pathway. There are many studies that explain the dynamics of this pathway and how its hyperactivation and dysregulation is also associated with cancer.